Okay, so the last time I had to talk during a session I chaired, I was not good at staying within the time. So now I set myself a siren. Let's see. Okay, great. So I'm going to just go to introducing myself. Just so you know, I work on a lot of different things. My background is math, computer science. I've been working in single cell immunology, biology for many years now, and have been moving into cancer also a little bit. And I don't have a background in physics. I ended up working with biophysics-y people by student initiative, let's say. So yeah, I think it's really useful, and I've been learning a lot. I think it's really interesting to understand how these different approaches are coming together. Right. But you'll see that I have become very practical over the years. And so a lot of this is very much data driven, and it's like, does it help or not? Kind of, does it let us do something a little bit closer to right or not? Okay, so I learned from the previous talks that I should start with my last slide so that if I don't get to it, you know what I meant to say. And so one of the things I want to say is that I'm going to be talking about RNA velocity, which we've already seen as trash. And we're going to talk about how to make it a little less trashy and why I think it might be really interesting in certain contexts, potentially. And this is sort of in the vein, like much of this work is really in the vein of, we know that everything we're doing is wrong, but maybe it's, still useful, even though it's definitely not the right model. It's noisy, challenging, and problematic for lots of reasons, some of which have been highlighted. There's many more, but hopefully we are dealing with some of those reasons among our within our group. And there's a couple that we haven't touched on that I'll bring up in our work. Let's see. Right. So two of the things that you'll see is that we found one of the things that makes a big difference is just focusing on the right cells and genes. And by right, I mean those that are at least related to a process that you actually want to infer the dynamics of. And I think one of the things that goes wrong, although I absolutely believe Brian's results that say that that we're confounding growth signals with other kinds of processes. I think one of the other problems that happens is that we're just confounding processes that involve different genes, different cells. And if we separate those signals, we get much better results. So that's one thing we learned. And another thing that Lior's brought up is that, incorporating a little bit more of the actual physical process of transcription. Here we use a bursty model of transcription that's very simple, but even that simple model helps. We're not as careful as some people, but it turns out even a bit of care makes a big difference. Tons of work I'm not covering here. As Brian also mentioned this morning, there's been like 1 million papers, and some of them do really nice work in terms of constraining the dynamics by some of which are very closely related to things people presented today too, about chromatin accessibility, protein, transcription factors, right? There's lots of constraints that we're completely ignoring here. Ideally, we'll solve all of these problems together one day, but right now we're just going to solve a couple. And then the thing that I'm really interested in right now is actually how to port this into a spatial context. Because to me, using RNA velocity to learn trajectories of cell state transitions is really only interesting in a few different sort of settings. One is when we just have very limited data, or when the cell states are super close and we don't have enough information to know what comes first. We don't have longitudinal information, we can't get it. And there can be a number of reasons for that, like can be patient data, or even just biologically can be hard. So those are reasons why it can be really helpful when sort of nothing else is available. But I also think potentially in the context of spatial data, having that sort of instantaneous change, right? Instantaneous transcriptional change, that direction, if we could infer that sort of reasonably in a spatial context, then maybe we can start to get better hypotheses about the tissue kind of components that create early changes, for example, transcriptionally. We can look at very early dynamic responses. So that's the idea. Maybe part of it will work, and I'll show you why we think it might. Okay, so I apologize in advance to those of you who came to the tutorial and were awesome. This is going to be a little bit repetitive. I was going to do something very different, but then Lior started with all these metaphors, and I thought I better include my metaphors. So I couldn't drop them. Okay, so one of the metaphors I use is, if I think about, I don't like to discretize anything. I think about the motivations of my students, for example, in a continuum. There are those who do, more laboratory experiments. They really care about solving the biological problem. And there are those who vaguely care about solving the biological problem and are mostly really interested in the math and the computation. And we all manage to talk to each other. And, we can think about, if we sort of can observe something about the states of these people, how could we, could we infer this sort of structure, right? The other question, though, is this a dynamic? Does this continuum actually represent something dynamic, right? That's not necessarily the case. There are times when, people don't shift too much, right? They come in one way, they leave one way, they're somewhere on this continuum, but they're not actually changing their position on the continuum. And so that's a question that we'd like to ask, is, are people changing, and if so, what changes them, right? And I think about that as kind of analogous to, say, a cell in a tissue context, right? So, for example, I have a very biological student that came from theoretical chemistry. And it turns out, yes, these people have changed a lot. Like, they are definitely moving along this. But if all I saw was sort of a single snapshot, I wouldn't have any idea of that being the case, right? They might just look like extremes. And so I started thinking about this in this context of the lead author of the work that I'm going to be talking about, who is jointly advised by Suri, who is a co-organizer of this, although he likes to hide in the back. And so Frank came in with a strong sort of physics, chemistry angle, but was interested in biology. And I asked, what if we could detect small, nearly instantaneous changes? What would that look like in this metaphor? Maybe it would look like not just Slack posts, right? But maybe even the drafts of Slack posts, like before they got up. You could think of that as like the untranscribed mRNA from the student, right? And his posts have more words. All of a sudden, we start to see an uptick in posts that have more words of immunology, cancer, tumor, and so on, and less of like AI models and embeddings and things like that, right? So that's an indicator that something might be changing, either about his interests or his motivation. And if we knew even more, that it turns out this uptick happened in the context of him working very closely with a tumor immunologist experimental student in my lab, then we might posit that it's by those interactions, them talking to each other, that we actually create this change, right? And we start to see this change in behavior. So we might posit that at least sometimes this is a dynamic process, and why it's dynamic. So that's kind of the model that I'm imagining, at least metaphorically, for what I'm trying to do. And the reason that would be interesting is, right, that starts to give us tools to potentially engineer change the way we want it, maybe, or at least to understand it better. I don't think any conclusion would be, definitive. And the other kind of useful part of this analogy is that, there are continuum that have very different kind of dynamics. There's height, in which there may be no dynamic relation at the age of, students that are in there. Facial hair is dynamic, but it has very different dynamics. And in fact, only a limited set of the population of students in my lab participate in that dynamic process. And if I want to learn those dynamics, it would be better if I focused on those students than if I focused on everybody, right? Or I'm just going to dilute my signal with a bunch of people who don't actually grow much facial hair. So that also becomes relevant, right? That we want to focus on the right signal. Okay, so how do we see, what is the actual biological thing I'm interested in? One of the major applications that I'm looking at right now is the tumor microenvironment. Coming from the immunology side, I'm particularly interested in the immune component, and how cells sort of either they arrive at the tumor, or they don't get into the tumor, or they become ineffective in sort of fighting a tumor, right? There's all different kinds of ways that bodies don't deal well with tumors. And can we sort of learn a little bit more mechanistically, or at least can we get some better hypotheses about what kind of interactions produce that, by actually getting sort of early changes? What are the early signals, and what kinds of say niches or features of niches provoke those changes? I think I already said most of these others. I'm also interested in lots of other sort of dynamic tissue environments with lots of immune components, like gut epithelium and so on. but I want to get to the rest of this. Okay, so we've already talked about velocity, but just for completeness, right? The original model is super simple. We've got transcription, we have splicing, we have degradation. We know this is wrong, but nonetheless, potentially useful. And the basic original model is a set of ordinary differential equations. Essentially, what really matters is the ratio between spliced and unspliced numbers of transcripts for each gene, right? And we sort of imagine that somehow we can find, steady states, and then we're looking for deviations from that, right? If we see more unspliced versus spliced, it means that we're ramping up production, and the other way would mean we're ramping down. Notoriously buggy, but if you can do it, right, then you would sort of imagine that you predict a future state, as Brian was saying, which is maybe a nebulous way to frame it in the first place, by sort of asking how your direction, your velocity of you being a cell, because I'm anthropomorphizing again, how the direction of a cell compares to its direction towards other cells in the population. And so that's how we get those arrows that everyone sees, right? We take an angle and compare it. The thing is, one other thing about that, though, is that this is very dependent on what cells are in the population, not just because of the inference, but because if you're moving in a direction that isn't represented, then that angle doesn't, it sort of looks small, even if you're actually going somewhere, right? So there's lots of evidence that these velocity vectors are a little buggy. But the idea kind of is sound, I would say. Maybe, at least, somewhat sound. Worth investigating. Okay. So two things among many that this sort of model ignores. One is the fact that there's not a steady transcriptional process, of just like producing at a steady rate, transcript after transcript after transcript, right? We know that transcription, sorry, I'm a little dry here, that transcription happens in bursts. And so, you could imagine that if you don't account for that, then you're going to kind of maybe attribute a burst of transcripts to upregulation when it's not really, right? It might just be not doing anything. And the other thing that is sort of maybe obvious to me and to people who maybe are dissecting big clouds and blobs of cells from using single cell profiles is that cells do lots of different things simultaneously. They have different dynamics. And so they may actually, you may want to kind of separate what processes you're actually thinking about. And some cells may be doing them more or less. And so if we could sort of learn that in a data-driven way, then you might be able to infer better signals. Now, in theory, you should do this all at the same time. What we end up doing is just, at least initially, we're just doing very simple, at least conceptually simple approaches to solve these problems independently. And even that seems to help a lot. So the data that we're using to kind of, that I'm going to use here, we do lots of other kinds of analyses, but is this very well-executed study that uses metabolic labeling of nascent RNA, which has come up a few times. It's one of the ways to actually check the time point at which at least some set of the RNA was produced and compare it to others, so you can get some actual real notion of the sequencing of production of the mRNA in cells. And so if you don't use the metabolic labels and you just take, say, a C-Velo, which is basically the most common, I would say, state-of-the-art method, you get a bunch of arrows that are going backwards. And it's not just the streamlines, like the inference is really all backwards. And that could be for many reasons. It could be also this growth thing that Brian pointed out. But anyway, it's definitely not doing what we expect, right? So the first thing that we did is basically try to do a kind of data-driven learning of the processes. And basically from the work that I've done, a really simple but very effective way of doing this is a kind of probabilistic non-negative matrix factorization. I know consensus non-negative matrix factorization is really popular. I don't like the consensus part of it, but I don't think it really matters, honestly. Any kind of dissection of the genes and cells into some sort of low-dimensional kind of program-centered kind of embedding will work. What's important here is that, right, we're not using a discrete cutoff, and that matters because that means that you can think about cells as having, as, you know, participating to different degrees in topics. And then we're going to do something kind of a little bit weird, which is I'm going to kind of pull a bunch of what I'm going to say are, you know, topic specific cells using some threshold, which isn't necessarily the way you'd like to do this, but you know, I'm essentially saying, okay, any cells that are involved in this process, but I really want to capture a dynamic range. So I want cells that are even doing it a little bit. I just don't want cells that have nothing to do with it, right? Because I would like to capture the onset or turning off of a process. And I would like there to be sort of bleeding between different topics to the extent that that reflects reality.
Can I ask you a quick question?
You can ask me a long question. Yeah.
It looks like you've concatenated the unspliced and spliced matrices.
Yeah.
Can you explain why you do it like that, for example, as opposed to doing them separately?
Yes. Yeah. We use the unspliced for the topic modeling because it's super informative for certain genes. You don't want to ignore it. It is also super useful to have them contribute to the same topic, so you learn sort of one uniform framework. It is a little bit crazy because they're also not disconnected, right? So it's a hacky, like I said, a lot of this is hacky, but there's a sort of rationale for it. Yeah.
Also, it looks like you're doing some LDA variant or something like that. It doesn't matter.
It is LDA. Yeah, okay. If you're depending on which version of LDA, you mean like latent direct allocation. Yeah.
Why don't you just do, why would you put a cutoff instead of just putting like a,
so I don't put a cutoff when I infer the model.
No, but why wouldn't you put a zero, like why wouldn't you put a discrete, like I don't know what you guys call them in statistics, but with some probability I'm involved in a topic and then a distribution after that, a conditional distribution. Like, I don't understand why you use the cutoff after the fact.
Oh, well, I mean, so we use all the software, you know, to do good latent inference of latent Dirichlet allocation, right? And then we're saying, okay, now for the RNA velocity, I want to focus on certain cells and genes and infer kind of topic specific matrices for those. So for the inference, it's easier to focus on whole cells rather than parts of cells, right? So it's a bit, but like ideally you would fit an end-to-end model, right? It's just this way I kind of know what I'm dealing with in the in-between. I don't have time because I have 3 minutes now, so I'm gonna hold on. I'm gonna be strict with myself 1 second. So I don't have time to go into this in too much depth, which is just to say that, you know, this is super useful for increasing signal to noise, right? Like suddenly you have a lot more unspliced and spliced kind of counts, right, in that cell population that you're trying to model, which helps you actually get some signal. And so we basically do very exact, you know, chemical master equation simulation to estimate the joint distribution of these unspliced and spliced counts and fit the parameters. And so this is a little bit slow. It's fine, actually, the way that we do it, but probably for going where we want to go next, you know, this will get reinvented. But it's all pretty straightforward, and you know, it does a good job of recovering the important aspects of the distributions. Let me say that much. And then we do topic-specific transition matrices, which are both super useful and a little tricky to interpret because they are dependent on the weights underneath the cells. But basically now you have sort of a single process, a transition matrix for a single process. And when you're trying to kind of get some kind of global view, one, again, sort of hacky thing that's also useful is to think about just using the topic weights that you had to begin with to integrate the transitions. So I take a step according to one transition matrix, depending on how much that topic is weighted. Okay, so if you do this, you don't use the metabolic labeling data, you get much better inference from this data. It works for many, many contexts. It does not solve all problems. We investigated sort of some other kinds of, statistical physics-y kinds of approaches for analyzing these that don't just rely on streamlines, which I don't have time to talk about. I also just want to point out very quickly that I can't go into this in detail, but we used this. I had another student doing another project where we were trying to understand and identify transcription factors that marked progenitor states for two very similar immune cell types. natural killer cells and innate lymphoid cell, type 1 innate lymphoid cells, which can be very difficult to tell apart. And the only reason we sort of think they're different is because sometimes they do different things and they seem to have different developmental trajectories. And so this is a case where it's super useful to have actual directionality, right? Because when you collect the data, they come together. It's very hard to get any kind of sequence of them. And the whole point is to sort of identify transcription factors that might let you actually just not produce some of them, right? Like to knock them out. So we were able to kind of identify this sort of dual progenitor population and this other population that is very specific and use bone marrow experiments. Okay, so in my negative 1 minute, I just want to say, I'm going to skip this. I just want to say that spatially, right, if you do a similar thing, so here this is from the top thing, let me just say, the top thing is single cells from a mouse tumor model of colorectal cancer that's been treated with immunotherapy. All I've done here is I'm just showing 3 topics that are all focused on the tumor-associated macrophages. Sorry for the acronym. That's all on the top. And so this is not how I want to do this, but if you were to just take a gene signature, gene score for those topics, project it into this space, you can see that some of them have very clear, this is, spatial transcriptomics data from the same tumor model. You can see that it has seems to have very clear spatial, regionalization, and some of them don't. And so the question that I asked is, if we do a velocity model in the single cell, we don't have the unspliced in the spatial, so it's really hard to get some kind of velocity model there. Can we actually see any kind of regionalization of the cells that are really changing? And you can see that there's sort of really specific cells that seem to be changing. Many of them are not changing that much. So we did kind of a dumb thing just to convince ourselves that this was worth doing right. The dumb thing is, we made a gene signature for cells at the early part of this kind of trajectory and a gene signature for cells at the later part of this trajectory. And then you can kind of color cells if they're high in both in the middle. And you can see that, and then we basically took those same gene scores, check that they don't show up in other cells, so they're not like highlighting other epithelial cells or something like that in the cancer model. And you can see that it sort of looks like they're spatially distributed, and this is sort of statistical as well, that there's a change as the cells are kind of going, as they're spatially distributed closer to the tumor core, they're kind of seem to be sort of going through this upregulation of this specific program. So that could mean that they're just the early ones to see, that there's a gradient of some exposure to something. It could be communication, but it suggests that there is some sort of regional manifestation of this change, which I think Brian was supposed to heckle me and be very negative, and I see smirks, so I'm waiting for that. Anyway, so that's my, I would like to do this right and understand if kind of having a better lens lets us understand more about how cells influence kind of short-term responses. And that's the group. Frank and Hope did the work, and Ruxandra collected the data. She's jointly advised by Tom Gajewski, who's a cancer immunologist at UChicago, and Bonna and Ben and Albert Bendelak all were involved in the NK cell ILC1 project. Okay, that's it.